So today me and my friend Yusuf Yıldız will talk to you about the fluorescence activated cell sorting, or FACS in short. Fluorescence activated cell sorting is a modality of flow cytometry, which is a powerful technique for the measurement and analysis of multiple parameters of individual cells within heterogeneous populations. For example, here you can see a sample of blood, and we can see that there is like a wide variety of different types of cells. We could need to sort those different types of cells or measure their size, granularity, or internal complexity, and that is exactly what is enabled by flow cytometry. Flow cytometry is a powerful tool because it allows simultaneous multiparametric analysis of the physical and chemical characteristics of up to thousands of particles per second. This makes the rapid and quantitative method for analysis and purification of cells in suspensions. This basic working principle relies on passing thousands of cells per second through the laser beam, one cell at a time. As cells pass through the laser beam, they scatter the incident light or emit fluorescence, which can be detected by optical to electronic coupling system. The gazer data can then be analyzed statistically by flow cytometry software to report on cell size, complexity, and phenotype. This figure shows schematically the primary components involved in flow cytometry. So, first is the fluidic system. Fluidic system presents the samples to interrogation by the laser beam and also takes away the waste. The lasers, they serve as the source of light for scatter and fluorescence. The optical system gather and direct reflected light to detectors. Detector convert the optical signals to voltage pulses, and the electronics convert these voltage pulses to digital data that is subsequently analyzed by software, flow cytometry software. Now I will talk to you about the fluidic system. So the purpose of the fluidic system is to transport particles in a fluidic stream to the laser beam for interrogation. For optimal illumination, the stream transporting the particles should be positioned in the center of the laser beam. In addition, only one cell at a time should move through the laser beam at a given moment. In order to achieve this, the sample is introduced to the central core enclosed by the outer sheath fluid, which is usually a buffer or some saline solution. The sample flowing in the center undergoes laminar flow. It means that it does not mix with sheath fluid, and also it undergoes hydrodynamic focusing. It means that as the diameter of the stream decreases, cells are confined to the center, as you can see from here. Hydrodynamic focusing is required because without it, the focus stream of cells would not be created, and analysis of single cells would not be possible. With hydrodynamic focusing, the cells flow in single file through the illumination source, called the interrogation point, allowing single cell analysis. So interrogation point is the heart of the FACS, and it is a point where the laser intersects with the sample, and it's also the place where the optic system detects light scatter and fluorescence. You can see from this figure, from this quick video, the interrogation point. You see that the laser beam strikes the sample, and also there is like detectors which collect the scattered light in different directions. That's the illustration of hydrodynamic focusing. Now I will talk to you about the differential pressure effect. So the thing is, the sample pressure is always greater than the sheath fluid pressure. The sample flow rate is controlled by adjusting the sample pressure relative to the sheath pressure using a pressure regulator. Flow rate is increased by increasing the sample pressure, which widens the sample core, but it means that the more cells pass through the interrogation point at one time. Conversely, lowering the sample pressure decreases the flow rate by compressing the sample core, thereby allowing fewer cells to pass through the laser beam at one time. A wider sample core can result in some cells passing through the laser slightly off the center and therefore intercept the laser beam at a less than optimal angle. Generally, higher flow rates mean lower resolution and are best used for qualitative measurements. Lower flow rates can result in greater resolution since most of the cells pass through the center of the laser and are typically used in applications where the high clarity is critical. Now, the second figure also shows that increasing the sample pressure increases the core stream diameter, which means that more than one cell passes through the laser light at one time point. So what happens when the laser beam actually strikes the cell? When it happens, majority of the photons will travel unobstructed through the cell. Some of those photons will diverge slightly, primarily via light diffraction, and create forward scattered light. Forward scattered light will form the majority of those photons interacting with the cell. And since the scatter angle is quite small, in order to detect them, the photodiode is placed in line with the laser path to collect forward light scatter and convert it to voltage pulse. Forward scatter can be used as a measure of the cell size. It means that the bigger the cell, the more light is scattered and the higher is the amplitude of the forward light scatter. There is also the second type of the interaction of the laser beam with the cell, and it happens when the laser beam passing through the cytoplasm, which is translucent, strikes some organelle such as nucleus or endoplasmatic reticulum within the cell. When it happens, the laser beam gets scattered at a larger angle and it forms the side light scatter. Relative to forward scatter, light signals produced from side scatter are relatively weak. A photomultiplier tube is used to measure side scatter because it's a more sensitive optical detector. In order to detect them, the optical is placed perpendicular to the laser path to collect light scatter in this manner. This video shows how different types of scatter are produced, and it also shows that the voltage pulse produced by side scatter interacting with the cell of lower granularity is lower in amplitude than the voltage pulse produced by interaction of laser beam with the cell of higher granularity. Now I will talk to you a bit about the photomultiplier detectors, which are the most commonly used detectors in flow cytometry. And in addition to converting the photons to photocurrent, the photomultiplier tubes amplifies the signal, and this process usually requires the application of a steady-state voltage to the detector. By application of the voltage, we can adjust the gain produced by these photomultiplier detectors. So how it actually works: when the incoming photon strikes the cathode, the electron is discharged, and it is focused towards a dynode, and when one electron strikes a dynode, 2 electrons are ejected each time one electron strikes a dynode. Finally, the electrons which are amplified, they strike the photoanode, and the photocurrent of larger amplitude is produced. Usually the multiparametric analysis is a routine. So how do photomultiplier detectors actually work? In photomultiplier detectors, photons of light energy enter through a window and hit the photocathode. If the photon is of sufficient energy, they will eject an electron due to the photoelectric effect. And when this ejected electron is focused on the dynode, 2 electrons for 1 electron will be ejected each time the electrons will strike the dynode. As a result, the number of electrons will be amplified, and by the time they will strike the anode, a photocurrent of a much larger amplitude will be formed. So the multiparametric analysis is usually performed by combining the forward and side scatter data, and this is used to distinguish different cell types within heterogeneous population. In order to form those graphs, intensity of two different channels are represented along the various axes. This forms a dot plot where each event is represented as a single point on a scatter plot. Events with similar intensities will cluster together in the same region on the scatter plot. From this figure, for example, we can see that the monocytes, we know that the monocytes will form the cluster in this region of the graph. And we know that, for example, this type of cell, we can see that it's like smaller in size and it has less granularity. For this reason, we can say that they are forming a cluster on a graph which is located both lower and to the left of the cluster formed by the monocytes. At the same time, this type of the cell represented at the top, it has a higher granularity than the monocytes, but it is smaller in size. And for this reason, the clusters they form, they are located to the left of monocytes, but higher than monocytes, because they have higher granularity but smaller cell size. And also we can see that within this cell population, the number of this cell type was much higher because there are more dots in this region than the number of monocytes. Although this type of analysis can provide us with some specificity, the higher specificity can still be made, can still be obtained with the usage of fluorescence. And the usage of fluorescence for the cell analysis will be described in more detail by my friend Yusuf.
Hi everyone, I am Yusuf. What is fluorescence? It is another parameter that can tell us more about cell structure and function. A fluorescent compound absorbs light energy over a range of wavelengths that is characteristic for that compound. This absorption of light causes an electron in the fluorescent compound to be raised to a higher energy level, as it can be seen here in the figure. The excited electron quickly decays to its ground state, emitting the excess energy as a photon of light. This transition of energy is called fluorescence. The range of which a fluorescent compound can be excited is termed its absorption spectrum. As more energy is consumed in absorption transitions than is emitted in fluorescent transitions, emitted wavelengths will be longer than those absorbed. The range of emitted wavelengths for a particular compound is termed its emission spectrum. One of the most common ways to study cellular characteristics using flow cytometer involves the use of fluorescent molecules such as fluorochrome labeled antibodies. In the figure, labeled antibody is added to the cell sample. The antibody then binds to a specific molecule on the surface or inside the cell. Finally, when laser light of the correct wavelength strikes the fluorochrome, fluorescent signal is emitted and detected by the flow cytometer. When a fluorescent dye is conjugated to a monoclonal antibody, it can be used to identify a particular cell type based on the individual antigenic markers of the cell. In a mixed population of cells, different fluorochrome can be used to distinguish separate subpopulations. Fluorescence detection. Well, how is this fluorescence information collected? The fluorescent light coming from labeled cells as they passes through the laser travels along the same path as the side scatter signal. As the light travels along this path, it is directed through a series of filters and mirrors so that particular wavelength ranges are delivered to the appropriate detectors. This video is very good. As you can see, when the particle passes through the laser, appropriate detectors are captured to signal. Fluorescence data is collected in generally the same way as forward and side scatter data. In a population of labeled cells, some will be brighter than others. As each cell crosses the path of the laser, a fluorescent signal is generated. A voltage pulse is created when a particle enters the laser beam and starts to scatter light or fluorescence. Once the light signals or photons strike one side of the photodiodes and photomultiplier tubes, they are converted into a proportional number of electrons that are multiplied, creating a greater electrical current. The electrical current travels to the amplifier and is converted to a voltage peaks, as you can see here in the figures. The highest point of the pulse occurs when a particle is in the center of laser. As the particle leaves the beam, the pulse comes back down to the baseline. All of the voltage pulses recorded and can be presented graphically. Two-color experiment. More than one fluorochrome can be used simultaneously if each is excited at 488 nanometer and if the peak emission wavelengths are not extremely close to each other. We need to look at the spectra of the two fluorophores to see if they are compatible. Alexa Fluor 488 and RPE are commonly used together. For these fluorophores, 488 nanometer light is an efficient excitation source. When excited with 488 nanometer light, you can see that the emission peaks for these two dyes are far enough apart so that discrete emission data can be collected. Compatible dyes such as these allow scientists to easily detect two colors from a single laser. The amount of fluorescent signal detected is proportional to the number of fluorochrome molecules on the particles. So if we analyze data from two-color experiment using scatter plot for discrete populations, we can comment on that in terms of quadrants. Cells with only bright orange fluorescence appear in the upper left quadrant. Cells with only green fluorescence appear in the lower right quadrant. Cells with both bright green and bright orange fluorescence appear in the upper right quadrant, and finally cells with both low green and low orange fluorescence appear in the lower left quadrant. In that site, multiple parameters are necessary to dissect complex biological system. We said that more than one fluorochrome can be used simultaneously if each is excited at 488 nanometer and if the peak emission wavelengths are not extremely close to each other. But how does the flow cytometer collect discrete fluorescence data for Alexa Fluor 488 and RPE? Filters are available that can capture the peak fluorescence from each of these molecules. 530 nanometer bandpass filter will collect most of Alexa Fluor 488 peak, and 585 nanometer bandpass filter will collect the bulk of the RPE peak. Using these filters, the proportional amounts of Alexa Fluor 488 and RPE fluorescence can be recorded for each cell. These peak emissions wavelengths are far enough apart so that each signal can be detected by a separate detector. You can see that portions of each emission peak here overlap one another. This is called spectral overlap. Now let's remember what was filters. Filters are normally defined by one of the two parameters, either the center point for a bandpass filter or the cutoff points for a short or long pass filters. This is typical bandpass filter specific for RPE. It has a center point of 585 nanometer and a width of 42. So it passes light in the wavelength range of 564 to 606 nanometer, which corresponds to the emission peak for the RPE. These filters used to resolve the peak include a 550 nanometer long pass and 610 nanometer short pass filter. One more important point regarding data collection is the use of a threshold. If every single particle passing through the laser caused the instrument to collect data, the data pool will be dominated by information coming from a very large number of minor particles, which can be seen here. To prevent this, a threshold is set. When a threshold value is defined, only signals with an intensity greater than or equal to threshold channel value will be processed and sent to the computer. On the histogram, the left side represents the small particles which is excluded. This means that the majority of the event are the cells of interest. In most applications, after a particle exits the laser beam, it is sent to waste. Sorting allows us to capture and collect cells of interest for further analysis. Once collected, these cells can be analyzed microscopically, biochemically, or functionally. Not all benchtop flow cytometers are equipped with a sorting feature. However, they can be upgraded to perform this function. To sort particles or cells, the cytometer first needs to identify the cells of interest, then separate out the individual cells. Once the population of interest has been identified on a data acquisition plot, a region is drawn around that population. A logical gate is created from that region. This gate is then loaded into the cytometer software as the sort gate. The sort gate identifies cells of interest to be sorted out of the stream. Different cytometers have different methods of capturing a particle of interest. For example, the BD FACS Vantage system electronics cytometer applies a voltage charge to drops containing a cell that meets the predefined sorting criteria. Positively and negatively charged plates are presented on either side of the vibrating stream. As the charged drops pass by the charged plates, the droplets are deflected to the collection tubes depending on the droplet's charge polarity. Now the questions part. First, determine whether the statements are true or false. And please pause the video, then check the answers. And here, fill in the blanks with appropriate word.
Those are the questions from my part. 8th question is a multiple choice question, so choose only one answer which you consider to be right. In flow cytometry, what is site scatter a measure of? What is the purpose of fluidic system in FACS? Choose only one answer which you consider to be right. Tenth question. Fill in the blanks with appropriate word or words. Maximum of 3 words can be used. Those are the answers for the whole questions. For all questions. Those are the answers for all questions. And those are the references that we used. Thank you for listening and your attention.